Assalamu alaikum. Good morning to all of you. This lecture is on CRISPR breakthrough in human medicine and crop science. Connects them to global food security by 2050 and delivers a message that can truly inspire scientists, students, and policymakers worldwide. This lecture I designed for this CRISPR, the gene revolution, healing humanity and feeding the future. My dear, I am Dr. Mushtaq, scientist in plant breeding and genetics. Good morning, distinguished scientists, colleagues, and visionaries. It's it's an honor to here to share this technology with great scientists, students, and fellow colleagues, my subscribers, and my listeners. But about the technology that is rewriting the code of life itself. When history writes about the 21st century, it will not call it is the age of machines. It will call it the age of genes. We are living in moment when we can edit the very blueprint of life, when human can correct genetic errors, cure diseases, and design crops that feed billions, all with a pair of molecular scissors called CRISPR technology. ये कंची की तरह होती है, कट करती है. What is CRISPR? First of all, we will understand what is CRISPR. The genetic revolution begins. My dear, let us start simple. Every living organism, whether human, animal, or plant, is built from DNA. Four letters: adenine, thymine, guanine, and cytosine, A, T, G, and C, forming the code of life. Forming the code of life, my dear. Now imagine if we had the power to edit that code. If we have power to edit that code, to correct a single wrong letter in a book of 3 billion letters. Of 3 billion letters, we can correct a single word. And by doing so, save a human life for increase a farmer's yield. That is what CRISPR does. That is what CRISPR does. CRISPR stands for clustered, regularly interspaced short palindromic repeats, a natural defense system discovered in bacteria used to cut the DNA of invading viruses. In 2012, two brilliant scientists, Jennifer and Emmanuel, turned this bacterial defense into a revolutionary tool, a precise, programmable, and affordable genetic editing system. And for that, we received the Nobel Prize in Chemistry in 2020. CRISPR in human health. First, we will talk about it. Curing the incurable. Curing the incurable. For decades, diseases like cancer, diabetes and genetic disorders were thought to be incurable, locked inside the DNA. But CRISPR has changed that belief forever. First, we will talk about cancer treatment. A cancer begins when our DNA mutates uncontrollably. Traditional therapies, chemotherapy, and radiation attacks the cancer but also damage healthy cells. CRISPR takes a different path. It goes to root of the mutation. In 2023, scientists used CRISPR to modify a patient's T cells, the soldiers of the immune system, making them to more intelligent and powerful to recognize and destroy cancer cells. This treatment, known as CAR T therapy, enhanced by CRISPR, has shown remarkable success in treating leukemia and lymphoma. Imagine instead of poisoning the tumor, We reprogram the body to heal itself. That is not science fiction, that is science in motion. That is science in motion, my dear. Diabetes cure. Now let us talk about diabetes, a disease that silently affects hundreds of millions. In 2024, researchers used CRISPR to correct defective genes in pancreatic cells, enabling them to produce insulin naturally again. In animal trials, diabetic mice were cured permanently, and in humans, early results shown reversal of type 1 diabetes not by drugs but by rewriting the body's code, DNA code in pancreas. See sickle cell and genetic disorders. In 2020, Victoria Gray, a young woman from the US, became the first person successfully treated with CRISPR for sickle cell disease. For sickle cell disease, here bone marrow cells were edited to produce healthy hemoglobin, and years later, she remains disease free. She remains disease free. This is not only treatment, this is transformation. Go beyond the form, that's called transformation. From healing humans to feeding humanity. Now, let us expand the horizons. CRISPR is not only healing the sick, it is feeding the hungry. It is feeding the hungry. By 2050, the world's population will cross 9.7 billion. And to feed everyone, we must produce 70% more food but with less land, less water, and harsher climates. And harsh climates. Traditional breeding cannot meet that demand, but CRISPR can. In plants, CRISPR acts as a precision breeder, faster, safer, and more accurate than any previous method. We are not inserting foreign genes. We are simply reprogramming nature's own instructions. Instructions, editing the crop's DNA to enhance yield, resistance, and nutrition. And nutrition, my dear. Number 5, CRISPR in crop improvement. Real world examples. Number 1, disease resistant rice and wheat. They are globally high demandable cereal crops. Rice feeds more than half the world. A single bacterial disease, blight, can destroy millions of tons each year. Scientists in China used CRISPR to knock out the susceptibility gene in rice, creating a variety completely resistant to bacterial blight without affecting yield, without affecting yield capacity. Similarly, wheat resistant to powdery mildew was developed by editing three genes in one step, something traditional breeding could never do so quickly. They take near about more than 12 years. Number B, climate resilient maize and tomato. At the University of Illinois, CRISPR was used to tweak genes responsible for photosynthesis efficiency, making maize more tolerant to heat stress. In Japan, scientists created CRISPR edited tomatoes rich in GABA, a compound that lowers blood pressure. A beautiful example of how plant biotechnology meets human nutrition. Number C, biofortified crops for nutrition security. CRISPR is also being used to enhance iron, zinc, and vitamin E stable foods like rice and cashew, fighting hidden hunger in developing countries. This is not just crop improvement. This is genetic empowerment for food security. Genetic empowerment for food security. Number six, my dear, CRISPR versus traditional breeding and GMO, genetically modified organisms. Many people confuse CRISPR with GMOs, but they are very different, my dear. GMOs add genes from other species. CRISPR edits the genes already present in the organism. It is like the difference between adding a new chapter to a book and correcting a spelling mistake in an existing sentence. This makes CRISPR crops more natural, faster to develop, and socially acceptable. It's breeding at the speed of light. Speed of light. It's breeding at the speed of light. Number seven, Vision 2050. The future of CRISPR. Let us imagine 2050. Hospitals use CRISPR to cure cancer, diabetes, and inherited blindness. Children are born free from genetic disorders. Farmers grow climate resilient crops that yield more with less input. And every nation, rich or poor, becomes self sufficient in food. With CRISPR, we will not only fight diseases but also rewrite destiny. The power of this technology lies not in machines or money, but in minds that believe in the possibility of transformation. We must go beyond form that's called transformation. CRISPR gives us hope that no disease is permanent, no famine is inevitable, and no future is unreachable. Unreachable. Number eight, that's very important. Ethical and social responsibility. Responsibilities. But with great power comes great responsibility. Editing life requires ethics and precise as the scissors we hold. We must never use CRISPR for vanity or eugenesis to create designer babies or other or alter human identity. Instead, let CRISPR remain a tool of compassion, not competition, healing, not hubris. Let science serve humanity, not divide it. Now, my closing words about this technology. The DNA of hope. Dear colleagues, every century brings one discovery that changes everything. That changes everything. In the 19th century, it was the steam engine. In the 20th century, the computer, and in 21st century, it is a CRISPR. It is a CRISPR, CRISPR. This is not just biotechnology, it is the biology of the future. It is the biology of the future. When I look at a DNA strand, I no longer see a molecule, I see a message that says, what says? We were designed to understand. We were designed to understand our own design. Our own design. As a scientist, we are not playing God. We are not playing God. We are partnering with nature to heal, to feed, and to sustain life. Let the next generation say that in our time, when hunger, cancer, and disease threatened humanity, science did not stay silent. Science did not stay silent. We picked up our pipettes, our dreams, and our courage, and we wrote the story of life. Thank you. Take care. Khuda Hafiz. I hope so that you like, share, and subscribe Halima Foundation Kashmir. Khuda Hafiz. I dedicate this channel only for those mothers who are alone, who are alone, who are alone. Please subscribe my channel. Okay, take care. Khuda Hafiz.